Let's take a look at the mechanism for the synergistic interaction between CIDO, or type 2, and CODI, or type 1.5, RAF inhibitors in cells with oncogenic RAS. This is exciting because RAF inhibitors have generally been thought to be ineffective in RAS mutant tumors. RAF dimerization is necessary for RAF activation and signaling. RAF inhibitors interact with two important motifs in RAF, the alpha-C helix and the DFG motif. Both the alpha-C helix and the DFG motif can oscillate between two positions, called in and out. RAF is active when both the alpha-C helix and the DFG motif are in. Two RAF protomers cannot both have the alpha-C helix in the out position. This is an important point, so remember it for a bit later. RAF activation requires recruitment to activated RAS. In the case of a RAS mutant tumor, there are many activated RAS molecules, so therefore RAF recruitment and activation is also at a high level. Active RAF signals to MEK. Our goal is to treat cancer by stopping signaling at the level of RAF. Suppose we apply a type 1.5 RAF inhibitor, also called a CODI inhibitor. When this inhibitor binds, it locks the RAF molecule in a conformation with the alpha-C helix out and the DFG motif in. The bound protomer is now inhibited and cannot signal to MEK. However, the other protomer cannot be bound by the type 1.5 inhibitor because, and this was the important point that I mentioned previously, both protomers cannot have the alpha-C helix out at the same time. The DFG motif on the unbound protomer is still free to oscillate, and therefore this protomer is still capable of signaling to MEK. The type 2 inhibitor locks the RAF molecule in a conformation with the alpha-C helix in and the DFG motif out. The protomer to which the type 2 inhibitor is bound is inhibited. Notice that the other protomer can still achieve either position of the alpha-C helix or the DFG motif, and is still capable of signaling to MEK. However, because of the flexibility in conformation, this molecule could be bound by a type 1.5 inhibitor. When we apply the type 2 and the type 1.5 inhibitor in combination, signaling from RAF to MEK can be fully inhibited. The effectiveness of this drug combination in reducing signaling from RAF was predicted by our model and also observed experimentally in two different melanoma cell lines. Finally, we show that the drug combination synergistically inhibits cell growth, both in a proliferation assay and a colony formation assay. It is clear that the combination of a type 2 and a type 1.5 RAF inhibitor has a strong effect in RAS mutant cells.